Okay, and then I'll share my screen. So hopefully everybody can see my screen now and I can see everybody here, so that's great. Okay, so good morning, good afternoon, good evening, everybody, depending where in the world you're joining from today. Welcome to the first day of the RDA's 22nd plenary meeting, and also specifically, welcome to the Harmonising the Future of Multi-Omics Data Standards Integration meeting. Which is part of the working group. It's quite a mouthful, isn't it? But you can see it on the slide, creating a multi-omics metadata schema standard reporting matrix working group. So I'm assuming that you're all here today because you have some interest in multi-omics or work in some kind of omics domain. So welcome, it's good to see you. We have this session today now, and we also have a repeat session next week on Wednesday for others to be able to drop in. So for those who don't know me, my name's Connie Clare. I am the Community Development Manager at the Research Data Alliance Foundation, but I've also had the privilege of working with this group since it was established in November last year. And there has been some great work that has been done so far by the working group, and I hope that we can share it with you and get some of your feedback on the next steps. So a few logistics just before we begin. So you'll see I have already posted links and Lindsay is also doing that in the chat to the working group page, so you can read more about what the working group does there. We also have a shared Google folder where we have all of our documentation shared. And I've also shared the link as well to the collaborative notes. So if you haven't already and you are just joining now, please sign in on page 1. It helps us to know who is with us today. We will be having time for discussion during the session, so you can add to the chat, you can raise your hand and unmute, and you can add to the collaborative notes. And we have another document that we'll share with you that we will be hopefully working on today. This is a Research Data Alliance plenary session, so we will be adhering to the RDA's guiding principles and code of conduct. And hopefully nobody here needs to report a breach today, but if for whatever reason you feel like you need to, we do also have instructions on the collaborative notes. I would also like to just mention, in case you are new to the RDA or not quite aware, we are transitioning at the moment to a new web platform. So this is our new working group page that you will see, and we've got various different tabs that take you to different aspects of the group. If you are an existing member of the Research Data Alliance and haven't yet been to the new web platform, this is just some admin really. You need to reconfirm your account, so you can do that via the link. And from now on, we won't be communicating via the mailing list, but we will be using the group post feature, which you can see here. It is part of the group's page, and that should send you a notification by email. Okay, so today's meeting agenda. I'm obviously going through the welcome and updates now. You'll see the link to the Menti in the chat, so you can do that after the session if you want to. Lindsay, one of the co-chairs, will be giving you a refresher or an update or introduce you to the working group and its end goal and our case statement. And then we'll move on to looking at how we could possibly select criteria for multi-omics standards. And then we're going to have some time for open discussion before wrapping the session up at the top of the hour. So I'm going to hand over now just so the co-chairs can say a brief hello. We'll start with Lindsay, if you want to just introduce yourself, Lindsay.
I'm Lindsay. I'm a computational biologist at Pacific Northwest National Lab in the US, and it's nice to meet you all.
Thank you. David?
I am David Mollick. I'm a computational biologist as well, but with the United States Department of Agriculture, Agricultural Research Service, Arthropod-Borne Animal Disease Research Unit in Manhattan, Kansas.
Thank you, David. And to Tim.
Hi everyone, I'm Tim van der Bossen, and I'm a postdoctoral researcher in bioinformatics, mostly metaproteomics, at Ghent University in Belgium.
Thank you. So those are the 3 co-chairs that if you want to contact as well after the session or have any follow-up questions, you can reach out to any of them there. So over to you, Lindsay.
Yes. So just a little background on the group. I'm not quite sure how many folks are in our working group quite yet or not from the list. But for a little background, we're really focused on aggregation of multi-omics standards. So it's really challenging to integrate different omics domain data types, metadata, and standards just across the board in general. And so I think our main goal is to illuminate what's out there, review the completeness of what's out there, and then present it in a way where people who are interested in building new developments have access to that, even if they're not an expert. So you kind of get that expert level knowledge or curation, even if you're not an expert or maybe you're a service provider. And so at the bottom end of it, though, is, the main questions is, why do we care about making our data fair? And how can we assist in helping people do that? And so this is somewhat part of the effort, right? So you have experts from, across the globe participating in different elements of genomics, but we're really focused on sequencing and mass spectrometry-based omics, some metabolomics, lipidomics, proteomics, et cetera. And so the very two that are highlighted on the right in the red box, these are the ones we're focused on right now. So we've begun our landscape review, and that is just an enormous aggregation of what's out there. And so we're also using fair sharing as a resource as well. I'm the Omics community champion for them as a side. And so the goal would be from this landscape, we'll have a really great breadth of knowledge of, what the state of the art is for developments, and then building a collection of those omics standard as a resource that can be iteratively updated for everyone who's interested to use it. So, and then going on from there, we're going to take that collection and cross-analyze it. So how compatible are the different standards across different omics domains? Can you intersect and integrate pieces of those if there are gaps in one versus another? And so I think there will be a really great set of collections and deliverables coming out of the group. And so we'd love to hear your feedback on that as well. So we can, it's perfect. So thanks, perfect, there we go. So this is just a quick screenshot of what our landscape review looks like. We have, of course, a landscape term dictionary to the far left, and I'll walk through this after this slide. And so what we've done is we've aggregated what those standards are, provided direct links to them if it has a fair sharing record. And we've added also into our deliverable, where does this fit in the data lifecycle? And so I probably could put a link to some of our slides that have a graphical overview of that. And so that kind of helps us also assess, all right, they have these standards, but where do they fit? Is it collection? Is it design? Is it reporting? Is it data acquisition, et cetera? And so as we populated this and filled these out, now we want to go through and build criteria to select these moving forward in our Fair Sharing Omics collection. And so we'll get to see a few examples of that across the different omics. And then, so in defining the criteria, if we have this large collection of what's out there, but in order to down select those and move forward with kind of what's at the forefront of the community and what people are doing and using. And this is aside from publications and repositories, right? This is really driven by the community and their developments. And so just some questions to ask as we go through these to select the to build criteria, but also select them. There's a few example questions in here to sort of assess and ask yourself, and we'll familiarize ourselves a little bit more. We have, I think, an hour set aside to go through these and start building examples. And so that should do it for the for that slide. So now I can probably share my screen, and if there's questions on this, just feel free to. We don't have too many people, so it's not hard if you want to. Okay, here we go. Can everybody see this?
Yes, we can see it. It's a bit small, but we can see it.
Oh, let me zoom in. That better?
Yes, it is better.
Okay. So I'm just going to briefly walk through how to sort of go through this. And the very first tab is the term dictionary. So throughout the genomics, proteomics, metabolomics tabs, you'll see some of this language being used. So here's a way to say, okay, I don't really know much about proteomics. What is it? Or I don't know very much about metabolomics. What exactly is that? So we're also using some controlled vocabulary for that to help. Some examples, this will link you out to searches in fair sharing. We've already kind of done that, and then also relevant tags. So if you're not familiar with fair sharing, it's a really great standards collection resource that links to other types of standards and databases. I can always demo some questions if you guys have some of that later. And then these specifically here are the types of standards that we're collecting in our tabs here. So reporting guidelines, typically metadata reporting guidelines, terminologies, model formats, so file formats, things like that, and identifier schemas. And that covers a large breadth of options there. And then as we go through and we're collecting the life cycles, you can come in here and also kind of look at what exactly does it mean when these apply to the analyzed component of a data lifecycle. So it's a lot to take in. I encourage you to explore it. The next one, we've listed some standard affiliations. So who out there in the community is developing a lot of these resources. And that kind of puts those communities on a platform. So we're not trying to reinvent the wheel and we're not trying to create new standards. We're really just trying to illuminate what's out there so that new developments can kind of build from these or build with them. The live curation summary is we've been breaking out into domain groups. And so with this, I've just kind of populated within each tab that you'll see to the right, how many different types of standards across these have been aggregated. So, and if you're curious, I just put the description of what is a terminology artifact. So this is kind of a nice way to pop in and just see who has the most different standard types or, who could use a little bit more work. So we'll go into an example here. So make sure I'm all the way right. Perfect. So here's where I'm just going to scroll to the bottom so you can kind of see what these options are. So here we're kind of filling these out. We've put in some drop-down selections to make it easy for folks to curate. So these, of course, are our standards. And the multi-standard application one is kind of like, well, what if it's an identifier and an ontology type? Like, what if it's, kind of a reporting schema? So sometimes there's some sneaky standards in there that kind of apply more than once. So that's why we have that option in there. We want to know, is it established, maintained, or in development? So is this a new standard in the community? Is it something that's widely adopted and maintained by a group, maybe like Genomic Science Consortium, GSC, I think. HUPO is a big one. And then where in the countries are these being adopted or used? And so here you can sub-select some of the different applications, like does this standard only apply to genomics or does it cover genomics and transcriptomics, epigenomics, et cetera. And so here are standard names. And these are all kind of been carefully curated. So if there's duplicates, it will find it. The acronyms, because most people refer to them as acronyms. And then here's our life cycle within G through all the way down to the pink. And so under these different selections, there might be something like when you're planning the data life cycle, would this apply to an experimental design or maybe a computed analysis after the fact? So here we have a sequence ontology. It might be used in a computational analysis design down the line. And so we select that. If we don't know, you can put NA, or if it could apply in any sort of application if you wanted to, et cetera. And so you can peek through these and see what some of these selections are. We really like feedback. There's things that are unclear in these. Not everything will always apply and some can be confusing. So that's kind of why we do it. To move on from there. So this is of course, our criteria box that we'll talk about later today. We go back through and sub-select these. And so these are also just some of the links, who's an affiliation that's either maintaining this or sort of driving the development there. So we want to include that credit. We have their home pages, the reference publication, if applicable to this standard, any source code, a fair sharing record DOI if it applies, help documents, related databases, standards, and then comments. And so it's quite a comprehensive review. So of course we have our proteomics and metaproteomics subselection, and then metabolomics and lipidomics here as well. Universal is kind of unique in the sense of these are standards that are often used, but maybe could apply across the board. So it's not pigeonholed to just one domain. It can be applied across domains. It's very cross-cutting. Most of those are going to be your obvious terminologies, identifier schemas, et cetera. So if anyone has any questions today, we're kind of trying to go back through and, like I'd said earlier, define from these lists, how do we want to select these moving forward to our omics collection and fair sharing? What are attributes or requirements that as experts or as a community we would feel safe selecting or suggesting? And so that's kind of what we're doing today for that. And then I will stop sharing And does anyone have any questions before we move on this? It's quite a lot, so I definitely encourage exploring it.
I have a question.
Yeah, I was going to say, Angela, you've got a hand raised.
Hi, I'm Angela Page. I'm Director of Strategy and Engagement at the Global Alliance for Health. I guess I think you alluded to this at the beginning, but it was a very it was a very packed presentation that you just gave in a very short period of time. So I feel like maybe what is the like end goal here? How is somebody supposed to use this? Is it merely to identify things out there, or is it like, how do you see people benefiting from having this information in this way? And then I think the second question I have is, what is the ultimate deliverable, this spreadsheet, or you just referenced something about pushing it to fair sharing? I don't know if that is the idea that this will all live on fair sharing eventually, or what's like sort of the end goal and how will people use it?
Yeah, so that's a really great question. And it is a lot of information. So this Google Sheet exists specifically really for the busy work that the group's doing, and we can do it in a kind of a collaborative manner, even after meetings subside, if people want to go back in the week and add to them. And so this, as it sits, will be available. I think we've talked about how could we share this in a machine actionable way? How could we share this in a way that makes sense for that particular purpose? Because we'll never destroy something that we create, right? But we want to sub-select the state-of-the-art standards moving forward from this into the Omics collection. So there's a little bit more machine actionability with that Omics collection. I could go on about that a little bit longer later if we have questions. They do have a really great backend and data model schema for some of those things. So it connects more than just the standards. It connects the organizations to resources, databases, et cetera. How someone could use this type of landscape review is, let's say you have a consortium project where you're building a multi-omics repository or database, whether it's proprietary or not. And maybe there's no one on the project that's an expert in genomics, but you're going to need the metadata requirements and domain requirements to aggregate and share and integrate that data, right? So this is also just not only a knowledge resource, but let's say you want to integrate both some genomics and proteomics data, and no one knows where to start because you're going to have reporting requirements, formats for both. And so this is a way where we have the community working together to say, oh, we have this for our proteomics reporting schema, but maybe we don't report as much information about sample information the way that genomics would need to have that. And so this is a way where, as we analyze these, we want to find those gaps so that you can merge and bridge across standards versus just recreating new ones every time a new project or initiative arises, right? There's so many standards and so many wheels that get rebuilt all the time. It's time consuming and it's a cost component, right? So the goal will be that it helps save time, bridge those conversations between experts, and drive new developments without reinventing the wheel, and really using those consortiums and those groups who are doing really heavy work to develop these, and give them that platform as well. So hopefully that helps. In terms of how we want to share things, always open. We're still kind of ongoing with those discussions. So yeah.
Thank you so much. Very helpful.
I don't know if Tim or David have any follow-ups on that at all.
Okay. No, that seemed about right. And it's indeed, there are quite a few standards, and our goal is not to develop like yet another standard, but it's more like to capture everything which is there and try to find out what a good solution to actually set up a multi-omics data standard. Like what do we need in the end? Like if you want to start such a project, what do we need? Not only data, also metadata. So it's not only, also metadata. To give you one specific example, I'm currently working as a metaproteomics researcher. I'm working now with the metagenomics community to merge those metadata standards. We don't have, if you're doing a multi-omics microbiome experiment, that you don't need like 10 different metadata standards to report the same thing. So that's basically one of the direct applications of that.
I have just a quick question. Is there anyone in the group, and you can just like raise a virtual hand or a hand in general, is familiar with fair sharing records and how they connect? Anyone? maybe it's worth it.
It seems a short introduction might be a good idea.
Yeah. Let me just share the screen and I can, what might help is linking out for like an example. So let's, if anyone has a particular preference on a standard, let me find one that maybe is, oh, okay. We'll try, we'll try this one. This is the minimum reporting standard for, sorry, avert your eyes when I scroll, sorry. So I put the fair sharing record here so you can link out and see this and we'll peruse. So the uniqueness about this is they collect records about repositories. Take a second. And the standards that we're also collecting. But at these pages, so they're defined by this is a reporting guideline. They have, the main link. There's always a maintainer, some subjects. But here is where you can figure out, all right, what funders or what organizations one might recommend this. So if you're funded, for example, I'll just give a base one, like by DOE, the Genomic Standards Consortium is an initiative that they adopt. So they have a lot of reporting requirements. And so they'll say, this is a collection that we would recommend our researchers to report X sequence information with or something. So it connects also to the policies. So there's, a few policies here that also recommend that, what other related standards they're related to and what databases use them. So there's a lot of content like that, some help information, contact, etc., who owns these or funds these publications. But the interesting thing is you can download this on the API side, but there is a relation graph here that is really interesting. And I don't know if a lot of people know how to navigate this one. And so this actually on the back end will show you every single other type of record it's connected to. And so the bonus about that is we sub-select from our landscape review in this collection. Each one of those records in there will have this, this graph. So let's just kind of zoom in for a second. So, oh, whoa, too much, sorry. Whoa, there we go. So here's kind of how they connect based on the lines, the colors, of course, whether it is a standard or so if you hover over this, these are all the other databases or standards is directly related to. So this is giving you that knowledge graph aspect of these relationships. And this is where that machine actionability comes in. So we'll get to that down the line. We have other deliverables where we'll incorporate this, but this is one of those reasons it's nice to build a collection here that is ongoing. It's a live collection for people. So, we're not just doing all this work and then it just dies, because that would be unfortunate, right? So we're really trying to think about how this really benefits the community with the least amount of redo work as possible. So hopefully that helps. And if everyone's curious, those are the links that are embedded in that landscape review if you want to explore those. Allison Lister, by the way, is the person to talk to you about fair sharing. If you're curious, she's around. I know she's on at this plenary as well. So, all right. So should we move to the selection criteria part? I think we're on time.
Yes, there's no more questions. We can move on. Do you want me to share the screen again, or would you want to walk through the document? Yep.
Did you want to take over, Tim?
You can continue. Lindsay is fine.
Okay. Just one sec. The reality of talking too much. Okay. So this criteria selection is kind of new and still developing. We don't want to just say, oh, we created this, this Omics collection of fair sharing arbitrarily, right? We have to have some sort of quality assurance in place. And so a couple questions I'll just read through to kind of give an idea to think about is as we go through, our landscape review and we look at these standards, are there any, if you're an expert in proteomics or genomics, are there any gaps right now in how you report your data? So for example, one group might really have great acquisition reporting formats, but is there enough information about the sample to really get a lot out of that reporting? So this would be, and maybe Tim can even touch on this too, as a real life example. So are there any other standards that might apply in a different omics group, like let's say genomics? they're really well formed in a lot of their sample reporting metadata guidelines, right? So is there something there that could be adopted in the proteomics community to just supplement an existing standard or not? So that's something just to think about if you're an expert in that field. Are there other standards being formally used that would fit the scope? So are there ontologies that could be adopted into your reporting guideline that maybe you don't have right now? Maybe it's unstructured text, which makes it hard for the machine actionability, things like that. And then are there new developments that maybe not everyone's aware of that extend or deprecate an existing standard? So not because it's outdated, but maybe it just doesn't fit the format or it's incomplete. And so moving forward, there's a new one being developed. Another thing is, something that's, I'm not quite positive, I can't speak on it to say that it's really well adopted is, the care principles are those things that could be adopted in some of these standards. Things like that. What would boost FAIR? And that's something to really keep in mind. What does FAIR mean for you and your community? It's very different for everyone, and it most definitely changes across disciplines. So just thinking in that frame of mind, kind of like where are the gaps? How can we make things better? And if you don't know, I think just exploring what's there and seeing if you can even make sense of why these are standards for some groups or not. I think getting out of the constraint of that mechanistic, oh, we use it because we put it at this repository. Oh, we use this because this is all I have to pass off to someone for my project. So thinking about things a little more out the box, how can we share more in an organized way to sort of build that FAIR component around the community? So that's just something to think about. And all suggestions are great. I think it's worth looking through the landscape review and then just thinking about, okay, if I was going to select one of these, what should some criteria be? Like one for me, I'll just throw it out there is the original development being maintained, right? So regardless if it's at FAIR sharing, regardless if we know about it, is someone maintaining a GitHub repository where that standard lives? Is like, does it have the ability to be updated? That's something that I think is really important, even including with ontologies. And so, in the Universal, there's been talk about the OBO Foundry. Their policy is it must be maintained to be a part of that foundry. Is it maybe that's a good idea to select some from there who belong to that policy or in that group? So yeah, so hopefully we can have open discussion today and hear from a few of you about what your thoughts are about those types of criteria selections. And so I'll hand it back to, I don't know what we're doing for this next part, if we're like splitting up or just opening it up in the doc.
Muted. We didn't plan any breakouts, but we can do that if you'd rather split up.
Or you can just, does anyone have any thoughts in general about some of these standards, or if maybe it's actually a good idea to know who works with standards on a regular basis or not? Multi-omics. So let's see.
So I'm going to raise my hand because I work often with data standards and metadata standards. So feel free to do the same if that's the case for you.
What do you mean by work with? I am working with a standards development organization, but I don't use standards for data processing or anything. I don't do stuff with data. I'm just on the standards development organization side.
I would say both. So either developing or using. The alternative, which is a bad alternative, is just reporting data in a non-standard way. But that's something I also often see in our field, unfortunately.
On my side, I'm working in a phenotyping platform, so we are doing some phenomics. So this one is not part of all the listed above. And so I'm from Finland, University of Helsinki, and we are trying to create some common data standards in the Nordic country about phenomics. That when we are talking about something, we are always trying to talk about the same things. So at least that's how we are tackling these standards, that we are creating within a field, trying to establish some common standards.
And do you also have the issues when you're trying to integrate phenomics data with either genomics or proteomics or metabolomics data, or is it something?
It's awful. So I'm not on this part, but I would bet because even within phenotyping platform, we have difficulty to understand each other.
Yes, yeah.
So I would expect that integrating other omics data will be a mess. And we are asked to basically expand the phenomics to metabolomics to other omics, trying to integrate more omics and to do some meta-analysis. And what, at least on our side, we are trying to use is we are using information system developed by INRAE, which is making the data fair, which is used to store and organize the data and making it fair for opening the science. So I'm trying to implement this for our database or our institution and to train some people to use it in other Nordic country phenotyping platform.
You mean from INRAE,
Yes.
the National
So
Research Institute for Agriculture, Food and Environment in
INRAE
France?
in France, yes. So
Yeah,
they have
okay.
developed a phenotyping information system called FIS, which is meant to store and organize multi-scale data.
And is that a way of reporting that is kind of accepted by the phenomics community, or is it something?
We start to push it to the phenomics community, yes.
Okay.
We try to push it at least. It's a very versatile information system, which is creating unique identifier for everything you are creating, which is following the MyAPI standards. I don't know if you heard about it. So it is following some kind of close relation with MyAPI standards and based on ontologies.
I mean, it makes sense, right? So the issues we're currently having with only, let's say, meta or like genomics, transcriptomics, proteomics can also be extended to phenomics. And I think that's also written down in our landscape preview. But to verify, I'm now looking at Lindsay and David so that it can be expanded afterwards as well, that other omics fields can join. I see David nodding, so I assume, yeah, that's
The
correct.
phenomics? Is that what you're specifically asking?
Yeah, that's our goal is
Okay.
not to develop something that only stands for the fields listed in the well, the white paper, but more like that it can be extended afterwards.
Yeah, phenomics is a super interesting domain actually because it is very heavily multi-omic integrated. So I would consider phenomics to be very much a part of this effort. While phenomics applies across the board for almost so many subject areas, it doesn't matter if you're biomedical, material science, environmental, it doesn't matter. It's such a huge part of the technology merging. So I would consider that very heavily a part of that. But just, I think sometimes we have to be careful how many omics things we're going to throw in there in the spread. But I'll say that phenomics is definitely a part of the effort here on this.
I think Volmar, was it you next, Volmar, that you had your hand raised, and then Halford and Angela in that order?
So with regards to working on standards or working with standards, I am a data steward working at the National Bioinformatics Infrastructure, and we do training for research projects. And when there's kind of plenty of different ways of describing samples, and I mean, these things that are common, you could use this. I mean, it would be easier if you had a common way of both describing things, the common vocabularies and common aspects of the experiments configurations, and then have a shared file formats that you could also kind of prepare your submissions in when you steward the data from the individual research projects into these common databases. So in those scenarios, I see that it would be helpful to, I mean, both have this overview of the different standards that you can pick and choose from, and perhaps also converging on something that could convince some of the repositories to adopt a common submission format for some aspects of the data, at least where it makes sense.
Halford, would you like to
Hi, yeah, sure. Thanks. First of all, I found what Silva had to say very relatable, because I work in the area of plant physiology, where we're integrating every conceivable omics. The latest omics I heard about the other day was shovel omics, which is the the soil microbiome around the root zone of a plant. Um, but the specific community I work in is this summer wanting to work together to try and harmonize and improve a whole bunch of interoperability and research practices, from are we physiologically measuring the same things that we think we're all measuring when we compare our data, through to how do we choose omics standards. And what strikes me as particularly important, this is now repeating a point that came up a few minutes ago, is that they kind of need to be extensible, because you need to be able to say, all right, I have a proteome, but it belongs to this context. It exists in relation to this phenotype. And to a certain extent, those exist in isolated standards. But as someone who's just starting to try and navigate, and at the same time help my broader domain community navigate the world of standards, figuring out how to integrate these ideas is not straightforward. So yeah, any kind of orientation that I can get out of this meeting, or not today, but out of the next 2 weeks, will be very, very helpful and very, very useful.
Yeah, I think to just touch on that super quickly, that's something that we're going to dive into pretty deeply in I guess it's technically the second deliverable, where we do the crosswalk analysis. So that will take things to the next level, where then you're going to dissect the elements inside that, whether it's like the content, structure, you name it, and then evaluate, what can be harmonized across those. So you can think about it like thinking about cutting standards into a couple slices and just seeing where there's some fit. And then that's a deliverable in and of itself outside of the collection stuff, is using the ones from the collection, taking those, now cross-sectioning them and saying, okay, if we dissect this, what can be flexibly adapted or extended? Because that is absolutely a continuous problem no matter what. And I think it's a labor of love for curating that kind of detail about things. So we hope to get at that specifically in the deliverables down the line. So this is sort of feeding into that next step. But we're not there yet. But it'd be great if you wanted to join it and be a part of that.
and share the process.
Yeah, of course. That's what's been kind of nice about the group, is you have this high computational expertise on the group, and then you have domain experts, and it only works when they work together. So the more diverse this working group specifically can be, I think that can be very well executed. So we're really trying to make sure that we maintain that level of talent on the group, because I think the outputs could be really, really beneficial for the community, and it is a big pain point.
Thank you.
So I'm sorry, I think this is related, but I just want to make sure. Like I said, I'm with Global Alliance for Genomics and Health, so I see a number of our standards on there. Not all of them. So I have two questions. One is like, is the landscape analysis done? And if there's stuff that we know about that should be added, can we send it to you to be added? Great. But then I guess, like, when Sylvan, you were talking about phenomics, my brain is going to human phenotype places, and that's completely different domain, obviously. And so when you talk about, the phenotype of a human versus like whatever you're doing in agriculture, like it's going to be completely different.
Yeah.
And so I guess that's another question that I have, is like that part of the domain distinction, and how useful, I'm not a scientist, so like how useful is it to have that like species overlap within this landscape analysis, because for some people there's going to be a lot of stuff in there that's like completely irrelevant. Maybe just at least having like a species column would be useful.
So I think if I ask this, if I'm right, so like in the landscape review, having a column of like sort of applicable species to a subject area. So I think of subject as like, if you're in microbiome or you're
Yeah,
doing,
exactly.
phenotypic disease or something like that. Those are definitely two different things. Yeah, I think sometimes on a side note, I think sometimes when you integrate those data, it's the ontologies that shed light on what your subject is, whichever you select. And so if you're building a resource, selection of the ontologies used to describe what's in the metadata structure that you're reporting, sometimes that is a dead giveaway. However, it's a good point right there because like what subjects does this apply to? And that's not a bad idea to put in there. And yes, please add to the landscape review. It is open for everyone, especially those if you work for those affiliations, definitely.
We can put them into the
suggest
spreadsheet,
it even.
or should I send it to you? Okay, cool.
Oh yeah, you can put it in the spreadsheet.
Okay, cool, thanks.
So perhaps to clarify a bit what we're currently doing with the metaproteomics community, because Angela, touched also upon the species column. So when we look at, for example, Pride is a proteomics data repository. So basically, if we want to publish our data, if we want to publish our data in a manuscript, then we need to submit our data to a proteomics repository such as Pride. And currently, so I'm working in metaproteomics, so I'm working in microbiomes, and we have as ontologies for species, we have human gut, we have human proteome, human metaproteome, human metagenome, gut metagenome. So people are just filling in what they think is best and what they think is most suitable. So we decided with the Metaproteomics Initiative, which is a community by and for metaproteomics researchers, we decided we wanted to streamline our metadata. And even in proteomics community, it's not that common. So in the proteomics community, we started a few years ago with the SDRF proteomics format, which we also saw from the metabolomics community, for example, was also used in transcriptomics. So I mean, it's a well-known format, but of course, it doesn't make sense to invent all the new microbiome metadata stuff. So we contacted people from the Genomic Standards Consortium, GSC, and we asked, hey guys, so what do you use for metadata standard for metagenomics? Because of course they have these lying around, apparently since 2011, which I was not aware of. So now we are identifying the overlap between SDRF proteomics and SDRF metaproteomics, basically, and everything which has the same meaning in SDRF proteomics and their format, which is the mix S, which is the minimal information about any sequence, something like that. We just kick it out with all the new stuff that they want to know about the microbiome. We put it in our SDRF file. You might think, that's not great because then you still have like two overlapping formats. But we also have to be realistic. If we come up with something completely new for the whole microbiome community, nobody will use it. So this is some intermediate step as we see. Ideally, we would like to go to a biosample approach where people, if they sequence or if they take a sample, like literally they take a sample, they list it, and based on that sample, what they took, if they do sequencing or they do proteomic experiments, then they will list the repositories from that sample together. Does it make sense what I'm saying? So you have your sample itself in a biosample database, like on EBI or NCBI. If you do sequencing metagenomics approach, you put your sequences and all raw data in ENA, then you can link that to biosample. If you do proteome experiment, you put it on Bright or any other proteome exchange repository, and then you link it also to biosample. So that would be regarding metadata, a possible end goal. This is only for metadata in microbiome. So I'm only talking about that because that's now an example I'm very familiar with. But you kind of have to, or I have the impression we have to also have realistic goals. It's good to have like a final goal, but if we're going to push it onto the whole microbiome community, nobody will ever care about it, I would say. Also not my words. So we discussed this in a workshop, I think 2 weeks ago or 3 weeks ago in Leiden, with experts from metagenomics, transcriptomics, proteomics, and metabolomics. And everybody was like, yeah, this seems like the best way to go for now. And later onwards, we can move on to the whole biosample approach. Unless you have now a much better idea, then I'm still happy to hear this, of course. And once more, this is metadata, not data reporting, which is something else, of course. But I see Lindsay unmuted herself, so.
Yeah, so I was just going to share this in the chat, and this is just like a one example. I think someone was like, how would we use this or something? So this is just one effort. So there, it's NMDC, it's a DOE-based collection, right? They kind of approach this as well. And I know that they did a lot of work on this, and this is just honestly one example. So their portal links information about multiple data type collections, and they're doing it by linking the standard metadata, actually. So they're using the same type of ENVO vocabulary to track both mass spec and sequence information. So they developed a new, they kind of developed their own standard reporting guideline by merging multiple types of standards. So in the parentheses, I think I linked to the data standards page, and if you scroll down, they try to incorporate things from multiple databases, and they're using Gold, NCBI, Massive. They're using the MIXS. They're using in a collection of ontologies. They're able to connect the mass spec to the sequence data, even through taxonomy, identifiers, location, et cetera. And so this is just one example of if you could use things from this collection to build a resource, but let's say your needs are different. You want to do something similar, but you're not in that subject domain area. You should be able to do something similar by subselecting something from these lists. And kind of like how Tim said, I know that they're working on development of merging some of those reporting standards with proteomics, and that sounds fantastic because that is missing, right? If someone wants to go in and use that data, they cannot because there's a level of information that's missing. So this is somewhat of an example. I think down the road, maybe we'll try to have some of these folks present. And this is really the goal of like, how do you adaptively or flexibly integrate multiple standards to fit the need, but people could still use that data because the standards are comprehensive and established. So that's something that's an interesting component. I know I'm not super familiar with all the resources. Tim might know more, and so will David, of other groups that are doing similar efforts, depending, what country or what funding source you have. So if that kind of helps put a picture to something. I don't know. Is this kind of what you're talking about, Tim?
Well, the NMDC, I think that's more for, honestly, I'm not sure. So I've heard of the NMDC and I saw what they're working with, but they have data from soil, from ocean. I think it's mostly environmental. So, for example, human gut will not be present there. I think it's mostly environmental. And they also it can also be used as a repository. But I'm honestly not sure how it links to them because I'm mostly working with human gut data, and I think that's mostly environmental data. Well, I think it's only environmental data to be pretty sure about that.
Yeah, there's a lot of interesting developments in the human gut. I'm not positive, but this is kind of a really great example of there's many use cases for this. So it's interesting to see how other people are doing it and what they're using, and then kind of getting some of the experts in those standards communities involved as well, so that there's more of like awareness of like what are people using. This is kind of interesting. I think I have one more example I could pop in if people were curious about just simple adoptions into a data model. Of course, I only know some, not all of them. Here's one by a consortium group for, they're doing more immune signatures with disease. But in their data model, they've kind of come up with their own standard reporting metadata stuff, but their selection of choice for helping integrate concepts is through ontologies. So on the right, they have vaccine ontology, disease ontology, et cetera. So there's a lot of initiatives that are doing a lot of things with either one type of standard only, or they're trying to merge multiple types. So like reporting guidelines with ontologies, or identifier schemas with metadata reporting guidelines. So you'll see a lot of that. And really, we're just trying to capture what are all those options, and then how the community chooses to use it is up to them. So yeah, I think maybe we should do that in a meeting, is just like go over some examples. That would be kind of fun. And have those people present. tell us about their pain points. So to, I guess, bring it back a little on the criteria selection, maybe some of the things or challenges that we're hearing are, how would you build criteria around some of those selections to kind of help with those pain points? Like what would help build trust in some of those selections so you could build something off of this list of standards? Um, and if no one knows, that's also okay. Maybe that's where the examples kind of help to come in. And if anyone has any other questions or examples or thoughts.
Walmart.
Yeah, so more soft kind of standards, are those in scope, like the STAR methods from CellPress? Basically, you have a table with some headings on things that would be useful to report, like the batch numbers, if you have them, and the product numbers for the reagents and things like that. And then some sections for things that you should include in the descriptions for making things more reproducible or replicable.
That's a reporting guideline, I think. I don't know the STAR methods, but sounds like a reporting guideline. As in, what would you recommend if you write a manuscript? What do you recommend to report? Is that correct?
Yeah, so it is something that you would include inside of a paper. It's text, so it's not coupled to a formatting standard that would be deposited in the repository. But I think some of the information that's asked for there, it's done in a way that is relatable to researchers that are not necessarily technology experts. And it does ask for things that are sometimes missing in some of the kind of more technical reporting standards.
So you bring up something super interesting about this. So the types of information, so if someone knows the STAR methods and wants to put an example in the paper, feel free. So one thing about STAR methods is it does cover a lot of the same stuff that's already in some of these domain standards when they report stuff. So a lot of the I know like a lot of mass spec has some reporting guidelines about like different reagents. I can't remember exactly what the standard that one is. Maybe Tim can refresh. But some of that does get captured already in these metadata standards that exist. I think the literature that's a great effort for the literature to help. It's really nice and easy to navigate when you're reading a paper. The one thing that's kind of a struggle with that though is it's still unstructured text. So even if you provide it in literature in that way, you could never actually go through and automate the extraction of that knowledge in that format. So I'm saying even like NER approaches or MLAI, like to extract that from a table is so much harder than it is to extract that from like a JSON file where you could have that populated. So there's kind of a balance, depends on what the end goal would be. But I would encourage, that might be an interesting activity in generals of the STAR methods or literature, reporting guidelines like that. How much of that is actually covered in a structured format in a domain that's just not being used? And so that maybe could give publishers a better idea of, okay, well, maybe we could suggest this format by, this domain initiative because it's exactly what we're asking for, except it's in a supplementary format versus a table or so who knows? I'd be actually pretty interested to hear from someone in that field of work about how they decide on those things because I'm not quite sure. But yeah, it's a good, interesting thing.
So, quick word of context, and I mean, since I do support researchers with depositing data in repositories, I mean, the ones that have recorded this information according to the STAR methods, it's really easy for us to kind of map them to the repository's requirements. But sometimes, I mean, you don't have the same precision in the things that the repository asks for, so you lose precision by going from the STAR methods into the reporting standards from the repository, which is sad.
Sorry, can you clarify that?
So, I mean, sometimes there's no controlled fields for adding the information that is reported in the tables of the STAR methods.
You mean it's not an ontology term?
Yeah, I mean, you could probably add your own ontology term there, but it's not one of the kind of indexed elements of the repository. So it wouldn't really contribute to increased findability. And you would be basically on your own in choosing, so which ontology should I pick the term from? I mean, if you want things to kind of integrate smoothly, you need some consistency in how you go from the information you have to how it's recorded and then indexed and integrated across repositories.
Yeah, that is kind of tough. So that's almost like assuming the authors would know exactly what the community needs for ontologies and they could select it immediately, but there's like thousands of ontologies. So if it's a little more established and structured, then yeah. But that's something that, I think I've seen a few times where they have a really great metadata reporting schema in a great format, has great fields, but they could be using identifier schemas like identifiers.orgs, queries, or even ontology prefixes or whatnot to build that out a little bit better, but maybe don't. And so sometimes I think, oh, I don't even know how they would approach that. So, but maybe they could go to this list and use it. What I would suggest. But yeah, I think I'm not quite sure which journals do that the best, if I'm going to be honest. So, yeah. I think the goal too would be if we build criteria even for ourselves, can we fill those gaps that maybe publishers wouldn't know or databases wouldn't know so that that can maybe be a discussion where it gets incorporated. Yeah. So, Tim and David, what should we do? What do we feel like we want to build on the criteria? Do you want to leave it open for discussion?
It would be good to maybe go over the criteria and to verify because few of you are working with, some of you are working with Stan. It would be good to check our criteria. And the most ideal situation for us is that you're extremely honest. If you think they make sense, let us know. But also if you think they don't make sense at all, also let us know, because we will use those criteria as a kind of a way to filter what we need for the end product. So definitely be honest about what you think. Lindsay, will you share? And
I can share if that's
Or
easier.
yes.
Yeah, I'll share the Google Doc that we started to work on then, if that's the right one. Is this one okay?
Yeah, that works. I think we give a little background and some examples on there too, if that helps. If people want to add their names to this, just at the very top, you can continue down. If you want to contribute or comment or whatnot, then that would be also great. So maybe if people want to just take a sec, if you're in the doc, just to just give it a quick read, at least over the top, so you kind of get a little more background on it. And then I want to say on page 3, we have under the references, there's defined standard criteria selection, and there's just one tentative example. So we're still building, but that's where we could potentially just start building in, adopting and extending ones that exist already that we know of or that we think is suitable for the cause, etc. So we'll just give you guys a couple seconds though to cruise through that. And feel free to just ask questions out loud. It's not a problem. And there are some links in that background for folks who maybe are not familiar with, the aim of the collections at Fair Sharing, so like what they do versus what a regular record would do, and then how the deliverables we're working on now support that, and then what we plan to do with them, down the line. So I think one that comes to mind for me, like as I go through that landscape review, I automatically already kind of know like, oh, I would definitely select this one, this one, this one. And then I try to ask myself, why would I select it? And it's really not because repositories only take it or, a certain publisher only takes it, but it's more like it comprehensively covers the most information I need to reuse that data. So sometimes that's actually what I think about half the time when I'm looking through these reporting guidelines is how well does it cover the sample information? How well does it cover the data acquisition information? How well does it cover um, the even like those nuances mentioned earlier, the species? Do I even know what instrument they used? Could I figure that out from this existing guideline? And so that's kind of what I think about. So in sequencing, I think there's always a couple go-tos. With mass spec, there's always a couple go-tos, right? And so I think one thing that's in common across those is that they're very actively maintained by a community. And so that kind of brings back in like these initiatives that are building, either they've created them and maintain them at a GitHub repository, and they're constantly updated and revamped. So that's kind of one thing. And so this tentative example was, all right, if we're selecting across the omics domain, so genomics, proteomics, and metabolomics, is it maintained by at least one community where if anyone ever needed to update or ask questions or fix an error on that standard, could that be done? Or is it just static? Like, has no one touched that repository in 5 years? Should be adopted or not, kind of a thing. So that was one for me that I don't think I've written down there on there yet, but is that it has an active maintenance at the origin. So there's a party I can contact about that. I could send an email to someone about that, like, hey, could you add this to the standard? Things like that. So, and that's something you can find out from our landscape review, actually. So the ones that say established, maintained, those ones I know I can trust will continue to stay up to date with sort of the community needs and standards. So that would be an example of what I would suggest for something on our criteria selection. I know others would might say like, okay, well, something's established, but it doesn't need to be updated for the kind of standard it is anymore. So it's fine being static. Go ahead, David.
Yeah, I think just on a sort of different note, one of the things that we think about a lot in this group is, whether or not the standard is used. And I just sort of been back in my head for a while. Use doesn't, I mean, like, I think use is an interesting concept here because, you can think about something being used by like every bioinformatician. You know, there's like a file format that every bioinformatician comes across on the daily. But what if I think there's an alternative here when we think about use, is what if a standard isn't widely used, but in the places that it is used is incredibly important to those specific places? Like there's an implied data structure, as an example, there's an implied data structure around, the NCBI, um, like metadata reporting on genome assemblies. But it's not particularly an open standard, but it's obviously very important to the NCBI. And I think we should be capturing that sort of relationship. I think there's some valuable knowledge in that somewhere. Um, like obviously, maybe, they shouldn't be doing that and it should be an open standard. But, you know, there you have it. It's certainly important to the field.
Sylvain, do you want to follow up?
Yes, so it has to be established, maintained, but what about the referencement, that it is easy to find when you are looking for it? You know what I mean? Or to browse it, or to find it, or to find which one to use. Some, I don't know, database referencing different standards from different omics to facilitate the identification of the most relevant standards that you want to use for your omics. Don't know if it makes sense.
So is there like an example you could provide kind of with what you mean by
No,
that?
I'm not sure. That's why I'm looking for where I can find this kind of referencement of selection of standards.
Okay. So it just has an established resource collection like on the web?
Yeah,
Is that
for
what you,
example. Yeah.
yeah. I mean, honestly, there's a few standards I know of that only exist in like one publication and they don't actually have a collection for it. It's just like reported in a table. I think you're just supposed to create your own format to adopt it. So that's not a bad one. I was just writing down, feel free to jot anything you want in this list that we're looking at right now. It doesn't have to be perfect. I think it gives us a start to have discussions. And so that's actually a really good one is there a source location on the web that this is housed, and I think that's actually is very important. Because I think we even encountered not very many, but a few scenarios where there might have been a publication in the 70s and that's it. There's actually no source for it. People just know about it. So you kind of have to have some way to be able to find it.
Looking at you, Fast A and Fast Q.
Yeah. No, we searched for
Technically
that
standards.
publication forever. And David, you found it. I couldn't find it. We were like, is that ever been published? And we couldn't find it for a very long time, actually. So I was like, oh, people just knew to do it, or they knew they needed it. People just knew. So it's really great word of mouth, though, on that standard. So trying to think. So one that I think about that I think is a sticking point a bit for a few of these standards is some of them exist because they're the first of their kind and they've just continued to be adopted. I think the one thing that I see kind of being a struggle as we move forward, especially in like the interoperability world, is they're still stuck in formats that don't work well. But that doesn't necessarily require that they reinvent the content in it, but might need to reinvent the format of how it's being reported. And so, depending on some of these criteria selections, it's kind of like, is it in a flexible format that either could be transformed or not? So I think we're really going to come across that in metadata reporting guidelines. Like, we can only use Excel for so long, right? There's a lot of complications with extract, transform, and load mechanisms, but that's really required moving forward with building new developments and also just building automated developments. So I think that's one I think about a lot.
Something which is machine readable, that you can extract information easily from it.
Yeah, you work on this kind of stuff, right, Tim? Do you have some thoughts on that one?
Yes. So it's always a difficult balance between being machine readable and easy interpretable and making sure people use it. You can't ask people to fill in a JSON file because they won't. It's difficult. So for us, for metadata, Excel seems to do the trick. Well, Excel, a metadata CSV file seems to do the trick, I would say. Because it's machine readable. So we have tools to parse it, and we have tools that creates those CSV files. And even if you open it, you can open it in Excel. So it's kind of easy, I would say. So I do think people will still use Excel for a while, unless we come up with like tools and software that just extracts it directly from like a database. Can be SQL, can be SQL, or can be something more fancy. But for the meantime, I think that CSV files and flat text files will do the trick.
Yeah, I think we've discussed this in our last group meeting is trying to we might need to use this criteria selection in table format so that we can address the terminologies as they stand alone, reporting guidelines as they stand alone, and then like all general ones. Because I think some of these problems might apply to just one type of standard versus all of them, right? So that's why I just put in there if it was in regards to like one specific type of standard or all of them. Yeah, because there's going to be less constraints on things like an ontology selection, but there's not going to be as much flexibility, say, with some of the reporting guidelines per se. So, just a side note. It's not an easy job to do the criteria selections, actually. It's not as easy as I thought it would be, or very straightforward. So, well, I guess I would ask too, with some of the stuff that you're doing, Tim, was there like discussion around what elements do you use and what elements do you not? Are you only adding to something that exists? You're not taking away from what you already have to replace it with something else, but you're just adding on to it. So are you extending it, or are you completely giving that standard a makeover, where you're getting rid of some things and adding new things?
So everything that is required for the metadata standard in proteomics, SDRF proteomics, will be the same. We will use the same standard, and they're using that standard also in single cell proteomics, where they have other things they will add to. They will use it for immunopeptidomics, where they have also other attributes they will add to. Same for metaproteomics. The only thing that's different with us is that we take additional stuff from metagenomics, additional attributes. So we were adding stuff. Most of the stuff we're going to add is, by the way, also optional. Nitrogen, like how many percent nitrogen was in the soil, like those kind of things. If you don't measure it, you don't have to report it, but it is there. If you're taking an ocean sample, what is the longitude, what is the latitude? That's important to know. If you're taking a human gut sample, what is the age of the person? These kind of things, because it's interesting metadata.
Yeah. I guess the one thing is, so if you adopt that much and you merge it, is it technically a new standard, or they're still standalone, but they're reported together in a different format?
I'm sorry, I missed the first part of your question.
So if you merge that much information, because those are, I mean, I know I fill out those sheets every once in a while, and they're a little daunting at times. So, but
no, I got the question. So, it is SDRF metaproteomics. So we will, as it a new standard. It's also readable. It's machine readable by our tools because it has the same format. The only thing is we have additional attributes.
Okay. So for those who are less familiar with attribute selection from these, I'll actually put that in there and you can kind of see. So those reporting guidelines are interesting. They work really well in that database. But there are like a lot of attributes you can select depending on what kind of research you're doing, right? If you're doing gut, if you're doing environmental, et cetera, et cetera. And so I guess my question kind of was like, is there, like, you would have the ability to just fill them both out separately. Like, let's just say they're two separate files, you fill them out, and then you provide them together, or it's one file, like it's where you've just put that information inside the new SDRF format. Trying to visualize kind of like how that would look, like if it's just.
There will be overlap between the metagenomics and the metaproteomics when there are two different files and we don't have a microbiome metadata file. It's the same because ideally we would have one, but at this moment we are not ready to do that. Our first goal is to make people fill in metadata standards and not to have like only one file that takes everything into account.
Okay. I just put links in the chat if people weren't familiar with what we were talking about, because I'm curious about that and that process, actually, that mindset of, okay, what do we choose and how do we format it if we adopt it?
When you are talking that you are linking data standards, it means that you are creating some vocabulary between them, some ontology shape. That one standard, so you have a plant which is part of a leaf, which is part of a plant which has a germplasm. So you are creating this kind of link and vocabulary between the different standards. So like I mentioned, we are trying to use this FEES ontology-driven database, which is based on this kind of vocabulary structure organization of the metadata. Your experiment is part of a project, so it is linked to a project. This experiment has a specific germplasm, your plant has a germplasm, so they are just linked together and they are linking everything together. I don't know if I'm clear.
That's a biosample approach, right?
Okay.
So you
I'm
have
not
a sample and you do different.
Mm.
And if you take a subsample of it, you can make a new
Yes.
biosample, which is linked to it.
Mm.
So those are all options indeed. However, and maybe that was your question, Lindsay, I'm not 100% sure if I understood correctly, but if we're having a metadata file for soil, it's a different one than we have for the human gut or one for oceans. So, but in the end, it's still a different file than we have for metagenomics. Same environment, but different file.
Mhm.
Yeah.
Does it make sense what I'm saying, or is everybody still following, or I'm happy to explain again.
Yeah, the static component of merging those two is actually the SDRF content. What's interchangeable is the kind of sequence sample level content that you're after, and like the MI, right? So it's like what sample type, where was its ecosystem or environment, isolation methods, et cetera, et cetera. But then on the SDRF side, you're capturing more of that technology depth domain information, right? Or is it it's a mix of both?
Both technical
Yeah.
and biological attributes.
Mm-hmm.
Mm-hmm.
And basically, ideally, and that's also something we discussed, ideally all biological attributes will be the same for metagenomics, transcriptomics, and so forth. The only technical differences will be whether it's like how many reads that you have versus like the technical genomics, proteomics, that's different. Which search settings did you use to search your proteomics files? That's technically only relevant for the proteomics one. Therefore, the biological ones should go into kind of a biosample and only listed over there. And then we should have one specifically with only technical attributes for the types of data.
Yeah. It's almost like
But still, and
proteomics
I can't emphasize this enough, like we still have to motivate people to report metadata in somewhat way. If we're going to ask them to, and now you have to do biosample, and then you have to do the biological attributes in the biosample, and then you have to create a project that you have to link, they will say like, whatever.
Yeah.
We don't care.
Yeah, I actually think about this a lot. So I would say that the proteomic community is on the market for biosample adoption, biosample standard adoption. And that's a scenario I'll give you where I came across this once. It was I think it was what was the same thing between this biomedical sample, like let's say it was like a human urine sample and a soil sample that both were collected in the same geological location and were ran on the same mass spec instrument. Technically, in a data preparedness world, if you were to document those well, you could build new insights off of something that shares similarities between both their metadata about how they were ran on an instrument and their biosample level information. And right now, those requirements kind of don't really exist. And so I think the thing that Tim is talking about, if getting people motivated to report more than kind of what's minimum right now would allow for much more insight driven science, one, and two, it's still something that's flexible. So yeah, it's really interesting.
I don't see people here from the PSI community. I don't think there's people, but that's a discussion we have as well often, is how flexible should we be in our standards. You can say like, oh, it's a standard, we have to be very rigid, but if people, we're still in research, so people find new things, so you should be able to extend it a little bit. Like how much room for interpretation do you leave? Preferably none, but it's also not always possible then to build something or a new instrument comes out or a new technology and there grows your standard. So you need to leave it also a little bit of flexible in a way. because yeah, we're progressing as a field.
Yeah. This is good conversation. This is some of the conversation that gets missed in our regular working group because we're just busy populating things and doing things, right? So I think we'll pass this back to Connie. I know we only have a couple minutes left. So did you want to take over, Connie? Thank you everyone for participating. It was great.
That's fine. Thank you so much, Lindsay. I'm also just keeping up with the discussion in the notes. There's been so much shared, and thank you so much to everybody for participating in the chat and sharing all the links. I've tried to capture as much as I can and put it in the notes, and please feel free to add to the notes, add to the landscape review document, and also add anything else to the selection criteria document if anything comes to mind. It would be great if we could see some of you in our working group meetings. So please do feel free to contact us if you'd like to join, or you can join via the web platform, and hopefully then we can send you the calendar invitations for our upcoming meetings. I don't have any more slides to share, but I will just share our contact details with you. They are in the slides on the last slide, but also in the chat there. So if you do want to follow up with any of the co-chairs, you can do that, or myself as a facilitator. But I think we can finish a couple of minutes early. Thank you again for your participation today. It's been really a productive discussion, and yeah, I look forward to seeing what the group will do next. So thank you very much. Enjoy the rest of the plenary.
Thank you all.
Thanks.
Bye.
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 Thank you.